Hello, everyone, and welcome to today's live broadcast, ELISA First Principle, presented at the Abcam Virtual Expedition, Support and Products to Advance Your Research Virtual Event, and presented by Dr. Ralph Paul, a Scientific Support Manager at Abcam. I am Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Abcam. For more information about our sponsor, visit www.abcam.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appear on the screen. We'll answer as many questions as we've had time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or just use that Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Click on the Accreditation button located at the promotional board at the bottom center and follow the process to obtain your credits. Please join me now in welcoming our presenter, Dr. Ralph Paul. I'll now turn the presentation over to him. Welcome, sir.
Thank you, Susie. Thank you for that introduction, and thank you to all the viewers out there today for taking your time and joining me for this introduction and review of the ELISA technique and discussion of some common questions and issues that arise when ELISAs are being used by researchers. The overview of the webinar today I will be talking about general principles that apply to all types of ELISAs, and then describing some variants of this technique, including the direct, the competitive, the sandwich, the SimpleStep ELISA, which is a variation of the sandwich, and then a discussion of what could go wrong and what kind of troubleshooting or observations you might make if there are in fact issues in the running of the ELISA. So ELISA, which is an acronym for Enzyme-linked immunosorbent assay, was first described by Engvall and Perelman in Stockholm and independently by Schneurs and Weeman from the Netherlands, with both groups publishing in 1971. Clear immediate advantage of this general technique is it does not rely upon the use of radioisotopes and all of the shipping, handling, and disposal issues that radioisotopes present to laboratories. Variations of this technique have been developed over time, and a wide variety of variants now exist to analyze numerous analytes and in a very robust, sensitive, rapid, and high throughput manner. The term analyte will be used throughout the presentation, and it can be used to cover the wide variety of target molecules that can be measured in an ELISA assay. These targets can include small organic molecules, hormones, and of course, very commonly, proteins. ELISAs can be adapted such that the target analytes can be measured when present in a wide variety of matrices, allowing for substantial flexibility in sample collection and type. Typical matrices are described here: plasma and serum, saliva, urine, milk, cell culture media, cell lysate, and tissue extracts of various types. It is important when considering the type of matrix that you are looking at to try to match the ELISA kit specifically for that matrix. One can find numerous ELISA kits for specific targets that have been designed to be used with a specific type of sample, and it's important to match those as carefully as possible, if possible. I will start today when talking about the discussion and looking at the color development part of the assay, which will be common to all of the variants that I'll be discussing subsequently. The color development steps all rely upon an analyte being immobilized in a well that you can see in the diagram here, and then being able to develop color based off of the amount of the analyte present, and it will be trapped in various ways on the bottom of a plate. A very typical format is a 96-well format that's diagrammed here, but there are numerous assays that have been developed for much higher plate numbers, including 384 and higher, as well as different types of holding vessels that can be measured by a spectrophotometer. And in all of the formats, there are also blocking agents present at the bottom of the well surface to prevent non-specific binding of the HRP-bearing antibody or moiety. These blocking agents can include detergents, bovine serum albumin, non-fat dry milk, whole serum, and fish gelatin, and these blocking agents can be specifically matched to the type of matrices or analyte that is being measured in the specific ELISA assay. Using a multi-well format allows for numerous samples to be analyzed simultaneously and compared within the same assay. In many ELISA kits, the plates can also be separated into individual columns so that the entire plate doesn't need to be used up in a single run. This allows a researcher to come back and run additional samples later once they have collected those samples. We do, however, strongly recommend that each time a sample is run, a standard curve is run in parallel on the day of that assay. It can be difficult sometimes to compare OD values from standards run on different days, and interpolation of data for unknown samples can be erroneous. Therefore, it is strongly recommended that a standard be generated each and every time. After incubation with the HRP conjugate and washing, the solution of 3,3'5'5' tetramethylbenzidine solution, also known as TMB for short, is added to the plate. Over time, a blue color develops as a result of the TMB acting as a hydrogen donor for the reduction of hydrogen peroxide to water by the action of the HRP enzyme. This color intensity will be proportionate to the quantity of HRP present in a specific well. In this example, on the left, we can see standard control dilutions in the left two columns assayed as duplicates, also a strong recommendation, at least, if not triplicates. In the other wells, we see varying amounts of analyte present, also assayed in duplicates. And although protocol booklets will typically give you a standard incubation time for color development, it is also prudent to observe the assay as the color development continues and stop it if it looks like some samples may be getting saturated or multiple concentrations of the standard curve may be in danger of also saturating their signal. One of the ways to avoid any issues with this is to do kinetic measurements at OD 650 at multiple time points, and if there's any danger of getting to saturation, the last data set that was collected where you get a nice strong signal with a linearity of the standard curve can be used as the final time point. Alternatively, the typical experiment utilizes a quenching reaction. And in this case, the quenching solution is a 0.16 molar sulfuric acid solution, which inhibits the enzyme and stops its function. And at this point, the assay is held in the state of color development, although it's now in a yellow color, and this can be read at OD 450 nanometers. This is an endpoint analysis. The color will no longer develop, and then you can get your final data from that, and you can see what that would look like on the left-hand side. So this has taken us through the color development process, which will be identical for each of the particular assays mentioned below, and when we go into the details. There are other assays that have been developed using, for instance, alkaline phosphatase, but the HRP enzyme reaction is by far the most commonly used for various ELISAs. In the direct ELISA format, the analyte itself is directly bound to the plate wells, and a single antibody is used to detect the analyte, and this antibody will have been conjugated to HRP already. It is the simplest of ELISA formats and requires but a single antibody. And what would be an example of where this kind of analysis might be used? In an illustration of a direct ELISA being used, we can see data generated which was used to characterize an anti-human kappa light chain antibody. In this particular experiment, a constant amount of IgG analyte was immobilized on the plate, and then there is a direct correlation showing increased OD measured as the concentration of the conjugated antibody that is being analyzed here is increased. What's important to notice here too is that it indicates the specificity, as when there is a lambda light chain antibodies immobilized on the plate, there is no signal observed, or very little at the high concentration. This indicates the specificity of the kappa antibody has been demonstrated with an immobilized analyte on the plate. This is a version of a direct ELISA. There are other formats which can be set up like this as well, and there are other reasons why somebody might use this kind of approach. It can be used, as we saw in the illustration, for antibody screening and characterization. It can also be used if one is developing expression constructs for recombinant proteins and wishes to look at the various levels of expression in the various constructs or growth conditions of the cells or bacteria, for instance. And if it's an assay that's used very commonly in a lab, it is a nice way to go if you can only need to run it with one particular antibody, and you can perhaps pre-make plates in advance of running it. The pro here is there's only one antibody necessary. It eliminates any issues that might be related to the presence of a second antibody, and it requires fewer steps to run. Issues might be if there's any problem of conjugating the antibody to the HRP and binding activity is lost, there's only one antibody that you are in use, and you may have to transition to a different antibody to try. Direct antigen immobilization on the plate can sometimes represent a problem area as well, in that you might result in higher background levels due to nonspecific material being bound, and therefore a simple antigen preparation is preferred, such as a purified protein or semi-purified protein in the case of looking at a recombinant expression construct, and this may take additional steps and time to accomplish. And the other issue is that with a single antibody being used, one may wish to have some flexibility in assay development that is more inherent in a two-antibody system like sandwich ELISA, where you can look at binding different parts of the protein of interest and define a larger or a pro-protein from a fragment. The next example we'll show here is a competitive ELISA. And in this case, the analyte present is binding to a capture antibody that is on a plate, and then you come back with binding of a kit-supplied HRP conjugated analyte. So you have the same analyte that's being measured is now also found as an HRP conjugate in the kit. And this presents a competitive situation. So therefore, the greater amount of sample analyte that was in the sample you applied initially, the greater is the competition effect with the HRP conjugated version of that same analyte. What this results in is a standard curve that actually will go downwards. That is, the higher the concentration of the analyte present, the more competition there will be, and the lower the signal will become. The type of analyte that is very commonly measured in an assay like this are small molecules or steroids, because they are not large enough necessarily to accommodate the binding of two antibodies in a sandwich-type ELISA, and it can be difficult to raise antibodies to some of those type of targets. A good example of this here is a competitive ELISA that has been developed for the analysis of cortisol, which is a stress response hormone. And as you can see in the standard curve, as you go to higher concentrations, the OD value goes down. In this type of format, it's very commonly used for small molecules, and you can also look at now, in this case, starting to, because you can pull the analyte away from its surrounding matrix, that you can use a number of different matrices that will more cleanly wash and yield a signal. Therefore, more complex samples can be done. Again, you only need to have one antibody for this. It's less sensitive to the matrix effect than, for instance, sandwich ELISAs, because you can more cleanly pull the analyte out, and you also develop good reproducibility. However, not all analytes are amenable to this format, especially very small molecules sometimes are not very good as immunogens, therefore it can be difficult to generate very good antibodies to it. And for the small kind of molecules that I described here, it can be much more sensitive than a sandwich ELISA and much more viable as an approach. The most commonly known of ELISA formats, the sandwich ELISA, requires multiple antibodies in binding to the analyte. And here we see the various components, the capture and detector antibody, the detector most commonly associated with a horseradish peroxidase. This can be a direct conjugation or through something like a streptavidin biotin association as well. And in this format, as you can see, the more analyte that's bound to the immobilizing capture antibody on the bottom of the plate, the higher the signal will ultimately be, and you have a positive correlation with OD. And typical data is illustrated in the next slide, where you see an ELISA set up for human peroxyredoxin, and you see the very nice dynamic range in the nanogram per mil levels and the standard curve that's generated with the control provided in the kit. The sandwich ELISA does allow you a lot of flexibility in choosing where you would like the antibodies to bind that are responsible for capture and detection. It can also allow you to look at these analytes in various types of matrices. The sensitivity and specificity, now that you're using 2 antibodies, can be tremendous, as particularly the specificity aspect. It does require you to get a very nicely matched pair of antibodies that will not inhibit one another. I know internally this is something we spend a lot of time working on, is trying to ensure that we get the sensitivity and specificity we want with antibodies that will not interfere with each other's binding. And it also requires, therefore, that your analyte is sufficiently large and accommodating enough to have 2 or more of such binding sites so that you can get clear recognition in the sandwich itself. So now I'm going to talk about a relatively new development in sandwich ELISAs that is termed the SimpleStep ELISA, that by adding yet another antibody makes the whole process much more efficient and quicker to use. And it's a 3-step process, and you will have your ELISA result in 90 minutes post-sample prep. If the sample prep takes you an hour or so, then you're looking at 2.5 hours for running the entire ELISA assay, much more rapid than the standard type of sandwich ELISA. In this sandwich ELISA, the capture antibody at the bottom of the plate is specific to a tag, and this tag is now found on the capture antibody for the analyte. So you add the capture antibody, the analyte matrix itself, and the detector antibody conjugated to HRP. These are all mixed simultaneously together in a single-step incubation, and the anti-tag antibody pulls out the entire complex. The rest is washed off, and the color development takes place. Therefore, you get a very rapid and sensitive assay being developed with very few issues being found in our experience in talking to researchers that are using these particular assays. This is a representative set of data for a human PD-L1, and you can see here a very nice dynamic range with high sensitivity and specificity. The assays are always developed with that in mind to be as good as or better than any other ELISA currently available for any particular target of interest. The pros and cons are really very much the same as they are for the sandwich ELISA itself, being a variant of it. And particularly nice for researchers is the fact that you can run one of these in a morning and have your data to look at, and perhaps talk to us about in scientific support at Abcam, or be able to display in a presentation later that day. So in terms of resources, as to what could go wrong or where you might find answers or points that might point you in the right direction, especially if you're not just buying an ELISA kit but perhaps developing an ELISA by scratch, is if you will Google Abcam ELISA information, you will be taken to a landing page that has resources that will take you through how to run certain kits, what kind of considerations to have when looking into designing an ELISA, and what are kind of some of the problems that you may encounter perhaps devising an ELISA yourself. For further information, we encourage you to contact one of our scientific support teams across the globe, and you can find the contact information here. And we are happy to talk to you about either one of our kits that you might be using, or issues that you might be encountering with that, or if you're just trying to design a very specific and unique ELISA for your own work, we can try to help you pick appropriate reagents and set up appropriate controls for your assay. What kind of things could perhaps happen is, what are some of the questions we will typically get and talk to people about? Do you have a good positive control? Do you have a known positive control sample, a biologic that has been in some other assay shown to have a good level of the analyte you're interested in? Or perhaps you have a recombinant protein or purified analyte that is separate from that provided as a standard in the kit. That's always a nice way to make sure that you're properly picking up a signal. And the other conversation that we get into is people running assays and finding that they don't get a sample giving a signal, and often there the discussion revolves around the nature of the analyte. For something like the cortisol analyte that I discussed earlier, that's a stress response hormone, and each sample may not have a very high signal or any at all, depending on the level of stress that particular patient may have seen, resulting in expression or release of that hormone. So it is good to have some idea of what to expect given the type of sample you're looking at. And then is this analyte going to be present within the range of the quoted sensitivity for the assay? Do you need something maybe more sensitive, or perhaps something a little less sensitive so you don't have issues with running the assay itself? So that concludes my set presentation, and I am now open for questions.
Thank you, Dr. Paul, for that informative presentation. We will now start our live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question drop-down box located on the far left of your presentation window. Type that question into the box that appears on your screen and click the Send button. We'll answer as many questions as we have time for. So let's get started. Let's take a look at our audience member questions at this time. Our first question, Dr. Paul, is, the standard curve I generated is not exactly like the one in the protocol booklet. Is that okay?
This is a question we answer quite frequently because, of course, people would like their standard control data to look just like we show in our sample illustrations in the protocol booklets. And this is not necessary to get a very usable assay. The exact OD values can be shifted depending on how long the sample is incubated, the temperature that the incubation is run at. We find in our labs that just a couple of degrees of difference can really affect the HRP activity and the saturation level of the particular standard curve. And in addition, one might be using a different spectrophotometer to get the readout as compared to the one that we used in the lab to generate the data in the protocol booklet. So as long as you have a very nice correlation coefficient across the data points, you do have a good dose responsiveness. That is, at lower concentrations, the OD is lower, and at higher concentrations, it moves up in a predicted manner based on the dilution that was used in the concentration of the standard analyte present. You should be fine interpolating data from such a standard curve and get reliable data from it.
Thank you, and I want to thank our audience members for their incoming questions. Our next question is a somewhat related question. Our audience member wants to know, my standard curve is fine, however, my samples are failing to give a signal. Can you help me understand that a bit?
Right. So this is not, again, not an uncommon issue that arises. So the standard curve is working. So that tells you that the assay is functional in your hands and that the appropriate dilutions have been made and that the enzyme reactions are all working well. So the standard curve is fine. The samples, however, sample prep is one of the biggest issues that we discuss with people in talking about ELISAs. And one of them is always, can I prep samples and let them be stored for a while, or I did store my samples for a while. It's always good to take a really good look at the recommendations for a given kit. We will often recommend fresh samples be used, prepared that day or earlier that day, if possible, as close to the time of running of the assay. We'll sometimes give recommendations that if you're going to stop prep, to stop at a certain point, freeze the samples at a very specific temperature, be that -20 or -80. And the other thing I always recommend to people, if they do have samples that have been stored where there may have been some degradation, is to prepare at least one fresh sample at the time of running the assay so you can compare and contrast that one to the ones that have, for instance, been stored. Another reason for this kind of effect is that the matrix in which your sample is being measured is also an issue that may not be well compatible with the particular kit that you are using. So that is an issue that comes up quite frequently as well. People may wish to be looking at a plasma sample, and it turns out that the kit that they have in hand was directed toward samples that are tissue lysates. And those will often use quite different blocking protocols and solutions to give the signal. So those are very common effects that are observed, but making a fresh sample, if you're dealing with samples that have been in any way stored for any period of time, is always a way to see if the assay is working, but may not be picking up your analyte in samples that have been stored for some period of time.
Wonderful, thank you. We have a final question, and I want to remind audience members that any questions not answered today will be answered via email. Our last question is, my samples are giving a uniformly high signal. Can you help me understand that as well?
Right, so this is kind of the inverse of the previous problem where you can have saturation of signal by your samples, and if, for instance, this is an analyte that is present at some abundance in all of the samples you're looking at, it may be that you're simply saturating the system, and what you need to do is make some dilutions of the sample before trying to interpolate data off of the standard curve. Typically, we will recommend also, in a situation where it's an analyte that might be found at constitutively high levels in the matrix you're trying to analyze, is to perhaps start with a smaller experiment to look at a dilution series of 2, 10, 100-fold dilution and see if any one of those will fall nicely within the uniform part or linear part of your standard curve. In many of the kit protocols, we will give recommendations as to dilutions that might be used or concentrations that have been found in a very standard set of samples. And from those concentrations that have been interpolated and the sensitivity of the standard curve generated by the kit, you can make some calculations about the dilution you may require to get your sample to fall within the standard curve.
Thank you, and thank you, Dr. Paul, for your presentation. Would you like to provide any closing remarks for our audience today?
Yep, sure. The ELISA assay is a very commonly used type of assay. The variants give you a number of different possibilities. I, by far and away, haven't discussed all the nuances that may play a role in the data that you're generating, or if you have some very specific ELISA requests based on a novel approach you might be using that may use principles of ELISA, even though your assay wouldn't specifically be described as that, I would encourage you to contact us in the scientific support group, and we're happy to talk to you directly or bring it to the consideration of our R&D folks to help you sort some of those questions out and perhaps give you some guidance for your work. Thank you for your time.
And thank you, Dr. Paul, for your presentation and for your important research. I'd also like to thank LabRoots and our sponsor, Abcam, for making today's educational webcast possible. Before we go, I want to remind all audience members that today's webcast will be available for on-demand viewing through April of 2018. You'll receive an email from LabRoots letting you know that this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We hope to see you again soon. Bye-bye.